It's my pleasure to introduce Sharon Giordano, who is the Herbert and Olive Stringer Distinguished Professor in Cancer Research and Professor of Medicine and Internist, Department Chair of the Department of Health Services, Research, Cancer Prevention, and Population Sciences, the University of Texas MD Anderson Cancer Center. Sharon?
Well, thank you very much. It's a real pleasure for me to be here today, and I want to thank the conference organizers for giving me the opportunity to deliver this lecture. So what I was asked to talk about is how we extrapolate data from clinical trials to the patients that we're actually seeing in our clinic and use the data from clinical trials to make decisions about real-life patients. And as I thought about this topic, there were kind of four areas that really came out to me. First, I think it's really important that we always remember that clinical trials don't always include representative populations. And because of this, we often lean on observational studies to show us real-world outcomes, both toxicity and efficacy outcomes. But these studies can be subject to bias. I think recently, with clinical trials that reported, we've seen several instances of studies that are positive studies and have statistically significant results, but small clinical benefits, which is a challenge for how we apply these to our patients, and situations where the control standard that was tested in the study is really no longer the standard of care by the time the trial reports. So I'll start with the basic premise of this, which is that we know that clinical trial participants aren't necessarily representative of the real-world patients that we're treating in our clinics. And this has been demonstrated in many, many studies. And it's only a small percent of patients that participate in clinical trials. In the US, that would be less than 5%. Minority patients, particularly Black patients, tend to be less represented, and older patients are very underrepresented in clinical trials. And not only are they underrepresented in clinical trials, but the older patients who participate in studies tend to be even less representative in that they're more active and fitter than the older patient population in general. Clinical trials tend to enroll more late-stage patients than early-stage patients, and those patients have better performance status and fewer comorbidities. They also tend to be higher socioeconomic status, and in the United States, more likely to have health insurance and to have good health insurance. Nationally, we see that women tend to be underrepresented in clinical trials. Of course, this is not relevant for breast cancer, and in fact, I think for breast cancer, we have a paradox where it's the other way around, where our male patients, although it's a small subset of patients, have historically been excluded from clinical trials of breast cancer. So this lack of representativeness of patients for clinical trials really brings up the concept of efficacy and effectiveness. So I want to spend a minute on this. So we think about an efficacy study. This is a study that's asking the question of, under ideal conditions, does the intervention produce the expected result? And so these are the questions that we answer with randomized clinical trials with strict eligibility criteria and close monitoring of patients. But we struggle when the results are not necessarily generalizable. Effectiveness studies, on the other hand, are really looking in real-world settings, asking whether an intervention works. And this encompasses many more issues, including adherence, costs of care, patients with comorbidities, and side effects, all of which impact the ultimate impact of a treatment. And these studies, although not as clean as a clinical trial, may have greater external validity. So I just put up a comparison of the randomized trials, which are really our gold standard for an efficacy study versus an observational study. So the randomized studies have excellent internal validity, they have strict eligibility criteria, very select patients. However, they can be quite expensive and quite time-consuming. In contrast, the observational studies have the downside of being subject to bias, which I'll come back to, but have excellent external validity, are inclusive of real-world patients, are cheaper and faster. So when we're thinking about how we apply clinical trial data to real-world patients and how we fill in gaps from clinical trials, sometimes we will turn to observational studies. The AHRQ has published a methods guide for comparative effectiveness research. And they have a structure of recommending that we think through two important questions before embarking on observational research. And I want to touch on some of the issues that this brings up. The first question is really, are there gaps in evidence from randomized controlled trials for the particular question that you want to study? And this happens, and it happens an awful lot. So sometimes randomized controlled trials are just not feasible or haven't been done. This can happen in situations where there's no funding, if there's a drug you're interested that's off patent and there's no support for a study. Sometimes it's considered unethical. I love the example of seatbelts. Like, there's no randomized study that shows that seatbelts improve outcome, but of course that would be completely unethical to ever randomize people to a seatbelt versus non-seatbelt study. Trials can be difficult to conduct for tumors that are rare, and certainly in situations where doctors and patients are unwilling to support randomization. That happens a lot in clinical trials of treatment versus no treatment. For example, I remember when we were studying bone marrow transplants in patients, it was really difficult to ever randomize patients because both the physicians and the doctors had strong biases as to whether they wanted or did not want the treatment. And there's also a few studies that have been conducted of new surgical procedures and new devices or new technologies. The second situation that arises commonly is that the randomized study really doesn't answer your question of interest. And this can be not including the population of interest, and we see this a lot for clinical trials on older patients, or those patients who have significant comorbidities at the time of their cancer diagnosis. The studies also might not have studied patient-centered outcomes. Increasingly, these are being incorporated into clinical trials, but many studies in the past did not really look at the patient perspective. Many studies just don't have the funding or the time to follow patients for an extended period of time, and this leaves gaps in information about the long-term side effects of treatment beyond just the acute toxicity during the period of the clinical trial. And clinical trials also can have insufficient numbers to study rare outcomes. So if there's a rare outcome where you may have only had one or two instances in a clinical trial, you won't really see that until the treatment gets disseminated out to the population at large. The second question, which I think is a harder one, is will the observational studies provide valid and useful information? And really the major issue here and the major challenge to observational studies is all sorts of bias. Selection bias, performance bias, detection bias, attrition bias, selective outcome reporting bias. I'm sure you can add more. But these are not randomized populations, so there will be inherent differences between the groups you're comparing. And I think it's really incumbent upon us to assess and, if possible, to address the risk of bias in the clinical context of the disease that you're studying, the data that's available, asking the question of whether the observational study can produce valid results. So I just wanted to show a couple of examples of how we've used observational data to try to fill in some of the gaps from clinical trials or when we thought that the patient population may not be representative. So this is a study I conducted with my colleague, Dr. Carlos Bercenas, and we were interested in looking at the toxicity of common chemotherapy regimens that were used for breast cancer. And so what we did was we looked at several different large insurance databases. We first looked at MarketScan, which is a US database of privately insured younger patients, and that's the blue bars. And we also looked at SEER Medicare data, which is the cancer registry linked with our Medicare claims data, and that's the yellow bars for patients that are 65 and older. The panel on the left is unadjusted. The panel on the right is adjusted for growth factors. They look basically the same. But what we found was that, certainly that older patients, again, the yellow bars, had a high rate of hospitalization from chemotherapy-related toxicity, and really quite a lot higher than you might have expected from some of the clinical trial data. And we also saw substantial variation across the regimens. So some regimens, for example, TAC, had almost 25% of older patients ending up in the hospital related to chemotherapy toxicity. A second example is a study conducted by my colleague, Dr. Chavez McGregor. And we all know that trastuzumab has associated cardiac toxicity. And the incidence of this really varies according to the definition and the study that was used. So from maybe 2 to 7% with monotherapy, 2 to 13% with non-anthracycline chemotherapy, and more than 15% with anthracycline therapy. In an analysis that combined data from 2 of the pivotal trials, the rates of symptomatic heart failure range from 0.6 to 2.9%, and the risk was highest in patients that were older, on antihypertensive medications, or had a baseline lower ejection fraction. However, with these clinical trials, again, less than 20% of patients were over the age of 60. And in the US, the average age of diagnosis is about 62. So more than half of the patients would not have been included on this study. And so to use data from both SEER Medicare and our Texas Cancer Registry data with Medicare for patients with a non-metastatic breast cancer treated with chemotherapy, and then looked at the rates of heart failure using billing codes. And the final cohort was just a little under 10,000 patients, of whom about a quarter received trastuzumab. And in this study, we found that there was about a 10% absolute difference in the rate of congestive heart failure between trastuzumab and non-trastuzumab users. And as you'd expect, the risk was highest in patients who received both anthracyclines and trastuzumab. And I think the point I want to make with both of these examples is that the toxicity that we can see when we look at treatments that are applied in the real world in a cohort of patients that were underrepresented in studies can be a lot higher than one would have expected. However, before you think this is the answer to everything, the challenge is really selection bias. And this is really the primary threat to the validity of observational studies. And what this is a systematic difference between the groups that are being compared that really arise from both either patient or physician selection of treatments or the association of treatment assignments with either demographic, clinical, or social characteristics that relate to outcome. So basically you end up with 2 groups that just aren't the same, and it's very difficult to take away those differences. And these biases can work in both ways. So on one hand, you can have a selection bias that would mean that patients with more aggressive tumors and poor prognosis are more likely to get treated. And so this can happen. I mean, some of these things we can adjust for, like we measure stage, we can take that into account, but we don't always take into account some of the more subtle clinical factors like lymphovascular invasion or other tumor characteristics that as clinicians we know are giving our patients a higher risk of recurrence. And this can result in an implausible finding where the survival actually looks worse in people that got more treatment just because their disease was worse. It can also flip the other way, though. It can flip the other way because patients that are healthier are more likely to get treated. So if you have a patient with a great performance status, no comorbidities, you're more likely to offer them intensive therapy than the patient with comorbidities. And how do we address these biases? So they're hard to address. I mean, I think careful study design is most important. We have statistical tools, including regression models, propensity score, instrumental variables, all of which can help address this, but not completely take this away. And this is just an example that I have to show you how hard it is to get rid of some of this selection bias and confounding. And this is actually an example from prostate cancer. It's not a breast cancer example. But there was a published study that suggested that men who had treatment for prostate cancer did better than men who were on active surveillance or observation. And so we went and took the same data set and reanalyzed the data and kind of mapped out the survival. And you'll see on the curve on the left, the blue line is men who had prostatectomy for prostate cancer. And they did better than men who had observation, which is the black line at the bottom. But then when we mapped out men who didn't even have cancer, nothing at all, the men who had surgery for cancer did better, had better survival than men without cancer, which is obviously completely implausible that cancer improves your prognosis. On the right, it's a very similar analysis, but then we looked by cause of death. So if you look at the overall survival, men who get treated do better. If you look at prostate cancer survival, men who get treated get better. But then if you look at death from heart disease, if you look at death from diabetes, if you look at death from pneumonia, in all of those, men who get treated do better. Obviously, treating prostate cancer doesn't cure diabetes or pneumonia. So this just keeps me very humble whenever we're looking at observational data, realizing that you can do a beautiful analysis of all these statistical techniques, but it's very, very hard to eliminate these kind of fundamental differences in patients that as clinicians we use when we're selecting treatments. So I would say that these observational studies can help address questions of effectiveness or toxicity in real-world populations. I mean, we can use them when we can't answer a question from a clinical trial or the clinical trial simply hasn't been done. They have external validity, but really bias is an ongoing issue, and we need more statistical tools to address this. Then I'd like to switch gears a little bit and talk about two other areas that I think recently have been on my mind as one of the challenges of translating data from clinical trials into my practice. One is the statistically significant but very small benefits, and the second is the control population not really being our standard. So I think the affinity trial is a nice example of a situation where we have a positive study, statistically significant, but a very small benefit. So if we look at the 3-year difference in outcomes, it's only about 1% with the addition of adjuvant pertuzumab. And if you calculate number needed to treat, which we just got a nice overview of in the previous lecture, it's 111. So you'd have to treat 111 patients to prevent one outcome. Another example of this is the ExteNet adjuvant trial, and this was a study that looked at adjuvant neratinib after patients received kind of standard trastuzumab-based treatment for HER2-positive breast cancer. Again, this study had a positive outcome, but if you look at the absolute benefit at 2 years, it was only about 2%, with a number needed to treat of 44. So I think both of these studies sort of illustrate this example that we have of small advances in our treatment that are important and maybe very important for our high-risk patients, but they're difficult to apply because all these treatments have toxicity, they have additional cost, and we're balancing it against smaller and smaller benefits and outcomes for our patients. And then the final point, again, is the sort of constantly changing standard of care, so that by the time we get the results from the clinical trial and have the outcome, the comparison group is not really what we're doing anymore. And so the ExteNet trial, again, is a really good example of a study that got caught in this. The treatment, the neratinib versus not, was given after 1 year of adjuvant trastuzumab-based therapy, but that's not necessarily our standard in all patients now. Would there still be a benefit after dual HER2-targeted therapy with trastuzumab and pertuzumab? And now with the Catherine data, what about after we had neoadjuvant chemo with trastuzumab and pertuzumab and adjuvant TDM1, would there still be a benefit after 3 different HER2-targeted therapies? And we just don't know the answers to these questions. The Catherine study is maybe a little bit less extreme example, but still has some of the similarities. And so this was the study that I just previously mentioned. So after preoperative chemotherapy, for HER2-positive patients with residual disease, the use of TDM1. And this showed a striking benefit of patients, but again, less than 20% of patients received dual HER2-targeted therapy, which is what we're doing in most of our patients. So again, I think this is, when I see patients in clinic, this is another issue I struggle with, of trying to take the clinical trial data that was designed in a very appropriate way, but then by the time you get the results, it doesn't exactly apply to the patients we're treating. So, in conclusion, I just wanted to highlight that there's really many clinical situations where clinical trial data are relevant, but maybe not completely applicable to our patient population. And whether that be because the patient populations are very different, it's an older patient population, there's more comorbidities, or the benefit's really, really small, I think this is something that we really struggle with as clinicians. And I think it's important that we as clinicians remain cognizant of the limitations of trial data for these reasons. And these are really the areas of uncertainty where I think a very honest discussion of potential risks and benefits and shared decision-making with our patients becomes critical. Thank you very much.